everyone. My apologies for the chaos today. I had some interesting car issues very early in the morning. It was very cold. It was very dark. But I have all of that taken care of, and now I'm going to get you your lecture on satellite cells. So we are going to be talking about mitosis today. And what does mitosis mean? Well, essentially it means that we are going to take a cell. Let's get a better marker than that. And we are going to send that cell through something called a cell cycle. And that cell is then going to split into two identical cells. And we call this process mitosis. And in skeletal muscle, this process requires a lot of signaling. And the muscle cells themselves don't divide. Their satellite cells do. And every time we exercise and every time we cause damage to the muscle and we release growth factor signals, that tells the satellite cells to be released, to divide, and then to differentiate into muscle fiber cells, and then to be attached to the damaged muscle. So we're not going to talk about cell division in a normal sense, which means that almost every cell in your body is always dividing and always re-replicating itself. But skeletal muscle cells are very different. And skeletal muscle cells, after a certain age, no longer divide. And that is something really important to understand. And that's why we have these satellite cells that will divide for the muscle fibers. And we're going to talk about that process. So skeletal muscle cells, as you guys know, are multinucleated muscle fibers. We also call them myofibers. Right? You guys are aware of that. And these fibers are, after a certain age, terminally differentiated, which means they are what they are. We're not going to make any more of them. So if we were to, let's say we were in our 20s and we had skeletal muscle, and maybe you're aware of somebody that has experienced something like this, unfortunately. Let's say somebody was on a motorcycle and they got into a motorcycle accident. And let's say the car had hit them and crushed their calf muscle between the car and their motorcycle. And what had happened is when that occurred, there was a substantial injury in the muscle, and that took a big chunk of muscle out of the entirety of the skeletal muscle, right? So what would happen is, after a certain age, when that muscle starts to repair itself, it won't be like other tissues where we can get a lot of repair and replenishing of that tissue. What would happen instead is, once this injury started to heal, we would get something that would look like this. So the individual that received that injury would heal in a manner where the muscle would look like this. And they would have scarring, right? They would have scarring. And they would have loss of muscle function here, and there would be this big divot in the muscle that you could see with the skin, right? You could see that, okay, that doesn't look right. There looks like there's a big chunk of muscle missing there, right? So we could even exaggerate it and kind of go like this a little more, right? Well, that's because the skeletal muscle was injured, it perished, and it can no longer be fixed, right? So as part of that injury, the skeletal muscle would release these satellite cells, right? Because the muscle is injured. And those satellite cells would come from the muscle, and then they would try to bind to existing muscle to try to repair that. But they can't replace the muscle that was lost. They could only add to it, not even significantly. So they would look like that. So essentially, the muscle can't divide. It is permanently differentiated, which means after a certain age, we have what we have and we can't grow any more. And that means that these muscle fibers, right, all of these fibers that we've talked about over the entire semester, that's what these lines are, these fibers, they can't divide. Or they can't go through what we call mitosis. But these satellite cells can go through mitosis. So we have these satellite cells. They can help a little bit, but the muscle cell can't divide like, let's say, skin cells or liver cells or blood cells. They can't form new fibers after a certain age. They will never divide again. So when muscle grows or repairs itself, it's not from the muscle fiber, it's from the satellite cells, which can fuse to existing fibers, right? So these satellite cells here, they will only fuse, right? They will fuse to the fibers that were damaged, but it won't be enough to replace the actual tissue that was destroyed, okay? So that's my introduction to this mitosis, right? So when we have mitosis, we have this division in skeletal muscle, this division of satellite cells. And the satellite cells can do some repair, but it's not a division or a replication of a fiber. It's just fusing to existing fibers. And this will happen in bad situations and in good situations. So for example, when we weightlift, and we cause micro trauma, right? That's the big word here. So if I go back and I draw that same muscle, right? I draw that same muscle and we have been working out, okay? So think about that analogy that I just told you with the motorcycle accident, right? That giant chunk of muscle that was removed from the accident. Well, in a situation like that, we can't replicate skeletal muscle and we can't replenish skeletal muscle fibers. They are already fully developed and they will be that shape and size throughout the entirety of one's life. But if we start to exercise, let's say we're doing squats and we start to get these trauma markers, right? We start to get these small tears in the fiber, which we call micro trauma, right? That micro trauma is very, very, very different than macro trauma, right? Which what we saw in the motorcycle accident, right? Somebody's leg was pinned between the motorcycle and the car, and then we had a giant chunk removed. That would be macro trauma, which you can see here, right? But when we exercise, it's quite small. It's micro trauma. And for the micro trauma situations, when those satellite cells are released from the muscle, let me change the color to blue, released from the muscle, these satellite cells will start to divide, mitosis, okay, divide, 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 divide. We'll have satellite cells released here. They will undergo mitosis, right, divide, 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 divide. And those satellite cells that undergo mitosis, they can then bind to these micro traumas and fill in the gaps and repair those injuries from exercising, right? But look at the difference between this injury versus this injury, right? It's completely different. And then what generally tends to happen is as we are exercising more and more, and we have this sequence happen more and more, well, then we start to get a little bit of growth in the muscle, which is hypertrophy, right? So these satellite cells are kind of helping the muscle get a little bigger, right? We're not developing new muscle fibers. We're just letting the existing muscle fibers get a little bigger. Thank you to the satellite cells. And now you can see that the muscle is starting to grow in size, which we call hypertrophy, because of these satellite cells, right? So if we don't have mitosis, we don't have satellite cells repairing the muscle, we don't have muscle growth, and we don't have muscle repair. So that is all super, super, super important. So what I want to do now is walk through the stages of this mitosis so that you have a better understanding of what is happening to these satellite cells once they are released. So again, we are going to have a cell. In this case, we are going to call it, I'm just going to put SS for SC, right? This is, let me get here. This is a satellite cell. The satellite cell was released from the muscle. Let's just say because we have been exercising. And the satellite cell, its job is to split its genetic information, right? So right here, we have the nucleus. I'm just going to put N for nucleus. So the satellite cell has to put genetic information into a nucleus here and a nucleus here, right? So you can see that this line is splitting the nucleus, okay? And then what's going to happen there is we have this cell nucleus splits into two, and then we are going to have the satellite cell split into two, right? So from one parent cell, we get two daughter cells, right? And these are called daughter cells because they're coming from one parent, right? So again, let me go back and just make it fully understandable here. Once we have a satellite cell released, I forgot the E, because of exercise, that satellite cell then has to be split into two. And then this satellite cell will have to be split into two. And then this satellite cell will have to be split into two, which means this nucleus will have to be split into two, right? And we'll get some of it in this cell, some of it in this cell, right? So we're putting genetic information in there, which means this satellite cell's nucleus will have to be split into two. We have to put genetic information in there, and then those cells can continue to split, and we can have muscle repair, right? So again, this is called mitosis, and mitosis is very necessary for exercise because we have to repair that muscle. And what this allows us to do is conserve satellite cells, right? So we don't want to use all of the satellite cells our DNA has given us. So we want to release a few at a time and let them go through mitosis and let them split so that we can reserve some of those satellite cells and not use them all because we need them over the course of our life, right? So what happens now is this mitosis process, they refer to it as threads. So mitosis, what that means is it means threads. And why do you think it might mean threads? Well, because it has something to do with what is happening with the chromosomes in your DNA. So I'm not sure if you guys know exactly what these things are, simply because you might not have had molecular biology, but I'll draw a picture, right? So mitosis means threads. And if you look at our chromosomes, right, we have 46 chromosomes in our DNA. So if we look at a nucleus, let's just draw a nucleus here. We look at a nucleus, let's draw it in, right, of any cell. It contains all of the genetic information, right? So usually when I show you guys what a nucleus looks like, I kind of draw it like this, right? We have the double helix, right? Double helix, that's how I always draw it. And then we have the nucleotides here. That's all of our genetic information. But what actually happens inside of the nucleus is we have we have 46 of these chromosomes, right? We get 23 from mom, and we get 23 from dad, and we can get them in any sort of assembly or variation, which means we might take more recessive genes from dad and more dominant genes from mom, or, you know, that's just a whole molecular biology lesson, but we're not going to really focus on that. But what really happens inside of the DNA is we have these chromosomes in there, and I'll show you a picture of this in a minute. And those chromosomes are created of millions and millions of strands of this DNA, right, like this. And those strands are wrapped tightly within these chromosomes. So all of this information, if you look at my little circles here, all of this information is DNA wrapped tightly around itself, right? And that is what actually is inside of chromosomes. So if we look at this next section here, these chromosomes are condensed nuclear material, and all of our DNA is in there. So just to kind of show you another picture of it so you can kind of get a better idea of this, is if we look at a cell, here it is, right? Here we go. If we look at a cell, and here is the membrane, and here is the cytoplasm, and here is the nucleus, inside of the nucleus is all of this chromosome, right? And inside of the chromosome, you can see that if we zoom in right here, it is double helixes that are wrapped tightly around each other, right? So that is what your DNA really looks like if we were to look at it through a microscope, or if we were to, oops, sorry about that, I don't want to do that. If we were to look at it on how it really looks, right? And what happens in mitosis is we have to take these chromosomes, right? We have to take these X-looking shaped chromosomes, and we have to split them in half, right? We have to separate them. And then half of the chromosome will go in one of the new nuclei, and the other half will go into the other new nuclei, just like I showed you here, right? We have to split it in half. We have to send half of the genetic information here, half of the genetic information there, and we split this X all straight across this line here, which means that some of the DNA will go into one new nucleus, and then some of the DNA will go into this other nucleus, right? So this is what has to happen in satellite cells as well when we want to split them in order to repair muscle. And I'm just going to show you this picture because I told you earlier that mitosis means threads. It literally translates into the word threads. Well, this is what it actually looks like when those chromatin, or these X's right here, right? When these things start to split, you could see how these chromosomes here, look at how they're acting like threads. And we call this mitotic chromosomes because you could see that they're going to go through this process of aligning in certain ways and then eventually splitting, right? You can see a split here, you can see a separation here, and then you can see how they get packaged into two new nuclei, all right? So I'm just kind of showing you that to give you a bit more information on why we call mitosis threads, okay? So at the end of the day, we're talking about how this genetic information coming from a single cell has to be split into two cells, which we call cell division or mitosis, and why this is relevant for skeletal muscle repair, because skeletal muscle is a very different tissue than other cells that we have in the body. All right, so let's look at this cell cycle business here. And again, this is coming from a satellite cell. I'm just going to put SC after muscle injury. Okay, So a muscle's been injured. We've been weightlifting. A satellite cell. Let's change this color. Let's just kind of keep it to, let's do blue. Satellite cell is released from the skeletal muscle. And now that satellite cell must go through division, right? It has to go through mitosis and be separated into 2 new cells. And again, that genetic information that is in the chromosome, which is in the nucleus, also has to be separated, right? So this must also be split in half, right? And we have 46 of these, right? Some of the genetic information is going to go to that new cell. Some of the genetic information needs to go through that new cell. And then this is how muscle is going to get repaired. So what's problematic about this now is that whenever we are dealing with DNA, right? Whenever we are trying to replicate DNA, we have to have a lot of regulation, because if DNA replication goes wrong, we start to get things like tumors and cancer, because tumors and cancer are a result of cells dividing uncontrollably and not being regulated, right? And this is how we run into carcinogenic pathology, where we get the development of these tumors that no longer listen to regulation or signaling, right? So if we have a hiccup somewhere in DNA replication and splitting of cells, we can have dysregulation of those cells being split, and we could run into complications, right? So what does that mean? Well, that means that this cell cycle business has to be highly, highly, highly regulated, okay? So we have a couple of different phases here, and let's just go through these phases. We have G0, G1, S, G2, and M. All right, so G0 means rest. So when you are not exercising, when you don't have a muscle injury, let's say you are just sedentary and you're not really doing anything, those satellite cells remain at rest. They have to be signaled to be turned on. And we'll talk about that signaling probably on Friday, okay? So when they are in G0, they are off, right? So let's say you are walking through your kitchen and you bump into one of the corners of, let's say, a countertop. And it's really sharp and you're like, oh, I hurt my muscle, right? And the next day you see a bruise is there where the countertop hits your thigh. Well, if the countertop that you bumped into was enough to cause a bruise, we know that bruise is the pooling of blood, which means that the tissue was damaged, that would turn the rest off, and now the cell cycle would be on. And that bruise is enough microtrauma to activate the satellite cells, because if there's blood there and that blood is pooling, which is what a bruise looks like, that means the tissue has been damaged, and that means that the satellite cells will go from off to on, okay? And so they will leave the G0 phase and go into the G1 phase. And you could see that the G1 phase is what we call growth, right? So when we are talking about this G1 phase, the satellite cells are released from their dormant stage, okay? So this is called a dormant phase, or a quiescent phase, or a quiescent phase. So they have kind of different names for this. So when it is sleeping, when these cells are asleep, they are in a dormant phase, or a quiescent phase, or a quiescent phase. And that looks like this. It's Q-U-I-E, So quiescent or quiescent. And that just means that these guys, these satellite cells these are Z's, they're fast asleep, they're counting sheep, okay? But when we bump our thigh against that countertop and we get a bruise, well, that tells these cells that sleep is over and it's time to wake up, right? So the satellite cells, they will leave this quiescent or quiescent phase, and they are now awake. And they need to move from this G0 to G1 phase, okay? I hope that makes sense. Now, once they enter into the G1 phase, they're going to do a couple of things. The G1 phase is specifically designed for growth. So the satellite cells become active. They're no longer sleeping. Now they must undergo growth. And what they will start to do is they will start to grow in size, and they will start to grow in protein production. All right, so let's take a quick second to kind of rewind for a second here. I think you guys all understand what is happening there. So I have a couple of things here for you that is showing you what are the main phases within this process, right? So in G1, we also call this the gap 1 phase, right? So I can put here gap 1. The cells will grow, they will produce proteins, and they will prepare for DNA replication, okay? So this is a preparatory phase. And this phase is, I call this the hungry phase, because the satellite cells are very, very hungry during this phase. They have to take in a lot of nutrients, all right? And they have to take in a lot of nutrients because they are going to become active, and they're going to need to grow so that they can separate, all right? If we look at the S phase, okay, S stands for synthesis. Okay, that's this phase right here. If we go from G1 to the S phase, right, we move into this phase. This is where the DNA becomes replicated. And we have to start creating identical copies of those chromosomes, right? So I showed you that chromosome here, right? We'll just color it in to make it a little thicker. Well, I told you that has to be separated. And then that DNA that's in that chromosome has to be replicated, right? So we have separation of the chromosome, of the genetic information, and then replication, because when this half here goes into one nucleus, right? It's going to go into one nucleus, right? Here's that half. Well, now we have to replicate DNA to build the other half, right? And when this piece here goes into another nucleus, right? Here's that piece that we just severed. Well, now we have to replicate DNA to build that other side so that we have full representations of that genetic chromosome, right? So that is going to happen in the S phase, right? So that's the synthesis phase where we are synthesizing genetic information so that both new cells have the exact same genetic material, right? We don't want to have any changes or mutations in genetic material. Then we're going to go into the G2 phase, right? Which is right here. Oops, let me get a marker that works. There we go. G2 phase right here. And if we look at that phase, this is preparation for mitosis, right? So we have more growth here. And then we're going to move into the M phase, which if we look at this right here, from the G2 to the M, this is the mitosis, right? So this is where the cell that has grown, replicated the DNA, and grown some more, right? It had to undergo like a lot of these changes in growth. Why? Well, because it has to take a lot of material with it when it separates. So this cell is now ready to be divided in half in the mitosis phase, right? So the satellite cell is now ready to be split into two, okay? So let's talk a little bit more about that so that we fully understand why the cell grows and what it needs to take with it in order to be turned into two cells. Let me just get this cleaned up and then we will move on. All right, so let's talk about this growth phase and talk about why we need to have this G1 phase. So let's talk a little bit about cell physiology, right? So we know that the basic parts of any cell are going to be the cell membrane, right? We're all aware of that. We've talked about that quite a bit. We have the nuclear information, right? And inside of here, let's just stay consistent. We have all of those chromosomes, right? And those chromosomes have to be split, right? Here's a chromosome. Here we have 46 of them, right? They have to be separated. And then we have this cytoplasm, right? Which is here, is this aqueous solution, right? This water that's in the cell, right? So when we split this cell down the middle, and we have to create a daughter cell and a daughter cell, well, all of this has to go with it, right? All of this material inside has to go with the cells. So we have to create the material before we even split the cell. And that's why this is called a growth phase, because we got to start producing a lot of proteins so that when we split the cell, both of the daughter cells get the same amount of material, that we have exact replications of this parent cell, right? So I'm going to put this here. These major components plus more must all be replicated and shared between the daughter cells, right? And it's got to look like this, right? We have to replicate, again, this DNA has to be split. The cell membrane has to be split, right? And it has to be given equally to the daughter cells. And again, this is what we call mitosis, right? So we have to have this G1 phase, or this gap 1 phase right here, so that this mother cell or parent cell, I'm just going to put parent cell, which in this case we're talking about a satellite cell, it has to grow, it has to create proteins so that when it splits, both daughter cells have everything that was in this parent cell, all right? So we call this mitosis, right? This is part of the cell cycle. So now this parent cell, or this satellite cell, has to start making a lot of proteins. And why does it need to make proteins? Well, it's got to have mitochondria. It's got to put mitochondria into both of the daughter cells. It has to have endoplasmic reticulums. That's what ER stands for, right? So both of the daughter cells have to get mitochondria and endoplasmic reticulum. It has to have things in it like peroxisomes and ribosomes, right? Or the cell won't function. So we have to put those into both sets of the satellite cells. We have to put enzymes in there, right? Or the cell won't be able to eat. We need all of these enzymes in there so that when we have sugar or fat or proteins or whatever, the cells can metabolize them and create energy, right? So these ancillary components, right? That's what I'm calling them, ancillary hardware, right? These organelles, they have to be created here in order to be placed equally in the daughter cells, right? Because those daughter cells are going to get put, installed with all of that information, right? You can see that this one, all of those organelles are going in there. And you could see this one, it's going to have equal amounts of organelles. And again, it has to come from the parent cell. Okay. So when we are in this G1 phase, this growth phase, that means this parent cell has to start making all of this protein, all of these organelles, because it can't split itself until it's been made, all right? So the growth phase is essentially a building phase, and the building is going to start occurring in the parent cell in this G1 phase. So when we leave this quiescent stage and we wake up because we have an injury and we move into this G1 phase, the G1 phase means that we have to start building all of this hardware, all of these organelles to start putting into the two differentiated cells. And we also have to start thinking about separating that DNA, right? So we have all that chromosome, which is here, right? We have these chromosomes that have to be split and placed into the daughter cells as well. So the parent cell, it has a lot of work to do before it can even think about dividing. And that work is going to start occurring in the G1 phase, right? The G1 phase in the parent cell. And in this case, we're talking about satellite cells, right? So I hope that makes sense. All right, so here's that picture again. Once more, we're talking about mitosis. We're talking about a satellite cell, right? This guy right here. Satellite cell, that's these repair cells, or they're also the stem cells of the skeletal muscle. In order for it to start splitting, it has to go through, leave the G0 phase and enter the G1 phase, right? And it has to start building. So here are those phases again. And let's kind of talk about this. All right, so that first phase. So we have this first phase, which is G0. This is the rest phase, right? I'll turn it this way so you can read it. And this is the non-cycling phase, which means the cell is sleeping, right? It's that quiescent or quiescent phase. It is in dormancy, right? Skeletal muscle needs a stimulus in order to wake the satellite cells up. And that stimulus is generally an injury. So now, in this phase, specifically with satellite cells, they are just sleeping and they are inactive. They are not doing anything. They're acting how a satellite cell should act. It is carrying out its normal role. It is sleeping. Why is it sleeping? Well, because we do not want to use energy unnecessarily. So satellite cells are in a very low metabolic state and in a very low transcriptional state, which means they are not dividing. Okay? So we need some sort of stimulus to leave this phase. And when we leave this phase, we are going to move into the G1 or GAP1 phase. Okay, so we got hurt. We bumped our leg on the table. Whoop, we have an injury. Or we went and did legs yesterday. We did some squats. We got some micro tears. Those satellite cells are waking up. We'll talk about how they're waking up on Friday. They are activating and they are mobilizing, and they are now going to enter a G1 gap phase. So that parent cell, that sleeping satellite cell that just woke up, it enters gap 1 of the cell cycle. Okay, so in this phase, let me just remove what I wrote there. In this phase, the cell will begin to grow. This is the initial growth phase. The cell will increase its size, increase its cytoplasm, increase its organelles, and start replicating proteins. I showed you that in the last picture. Okay, we have to start growing. We have to start building. And whatever we build has to be split equally from the parent cell to the daughter cell, right? So whatever we start to manufacture has to be shared equally before this cell can split in half. All right? Sorry about that. Let me clean this up because I don't want marks all over this. All right. So something else is going to happen here. We are going to have lots of protein synthesis, which means we are going to make a lot of these proteins. We'll have large productions of those things I was talking about, ribosomes, enzymes, structural component, and we will start preparing for DNA replication. Okay? We do not want to replicate DNA until we have all of the proteins that we need in the cell. Okay, so now what is going to happen here? Well, I told you that this is the hungry phase of the cell cycle phase. So that cell, if it wants if it wants to start making proteins, if it wants to start doing DNA replication, well, it has to increase its use of substrates. It's a hungry cell. It has to build. Just like if you want to build muscle, you have to increase calories. Well, if a satellite cell wants to replicate and build all the proteins its daughter cells will need, it has to increase its consumption of substrates. So it will increase glucose uptake, it will increase fatty acid uptake, it will increase amino acid uptake. It is bringing in all of the nutrients it needs to build things like enzymes, and to build things like mitochondria, and to build things like ribosomes, and to build things like peroxisomes, all right? So it will increase that substrate material. And why? Because it's going to need a lot of energy. It's going to need a lot of ATP to make all of this happen. So we call this biosynthesis, right? So the cell is creating or synthesizing new biochemical material, and it's going to require a lot of ATP to make that happen. And it's going to take some energy for that parent cell to enter the S phase, okay? So it has to increase its consumption of macronutrients in that G1 phase. It has to create ATP in order to enter that S phase. So once it has done everything it needed to do, you could see it has increased in size, right? Bigger, right? It has resynthesized all of the material that it needs in order to start dividing. It has all of the ATP that it needs in order to move into the next phase of the cell cycle. Well, then that parent cell will move into the second phase, which is the synthesis phase, or the S phase. So in order for it to move into the S phase, it has to undergo a checkpoint. Okay? And what do I mean by that? Well, I just mean that there are regulators, there are enzymes that look at what the cell is doing, and it makes sure that the cell is producing what it needs to produce before it goes into the S phase, which just means that that satellite cell needs permission to go into the S phase, right? And why does it need permission? Well, because if we look at that first phase, right, we know that that biosynthesis phase, we have a lot of transcription and translation happening, which means the DNA of the satellite cell is creating a lot of protein. And when DNA is constantly creating protein, it can undergo damage. It can undergo mutation. It can make mistakes. And if we make a mistake, if there's any mistake in the DNA, that mistake will transition into the S phase. So if the DNA was damaged because of all the protein production that was being done, well, that damaged DNA will now move into the S phase, and we will start replicating damaged DNA. And that's the worst possible thing we can do in any cellular environment, because that is exactly what causes cancer. That is exactly what causes mutation. That is exactly what causes tumors, when we have the dysregulation of genetic information, and then we have replication of that genetic information, right? So what happens is, before we move into that S phase, we have this checkpoint where we will say, okay, are we going to pause or are we going to proceed? And there's these enzymes. You don't need to know what it is for this class, but they're called cyclins, and they're called cyclin-dependent kinases. And these proteins regulate this transition. They're like watchdogs. They make sure that before the cell moves into the S phase, it has created enough protein, it has done what it should have done, and it's not carrying any damaged DNA or any mutated DNA. Because when we get into the S phase, we are going to start replicating the DNA. So if the DNA is damaged, that's where we run into problems, right? Now, if the DNA is damaged, right? If this DNA in this parent cell is damaged, and then that cell is allowed to go into the S phase, allowed to go to the G2 phase, and allowed to go to the mitosis phase, Well, that means daughter 1 and daughter 2 will also carry that DNA that is damaged. And if the DNA is damaged, daughter cell 1 and daughter cell 2, they can't perform their jobs. So they can't fix muscle because the genetic information is damaged. So this one can't fix muscle either. So now we can't have repair of muscle tissue, and that's problematic. All right. So having said that, we're now going to talk about the S phase. All right. So let's just say everything is running perfectly. Everything that needed to be built in the G1 phase was successfully built. There was no damage to DNA. The DNA is okay. We get to this checkpoint. We are given permission to proceed. And now we can go into that S phase. Okay. And this is where you should be like, oh, that makes a lot of sense why we have to have checkpoints. So when we get into the S phase, this is where we have to start breaking up those chromosomes, right? So we have to, let me get a different color here. Let's get something that will pop. Let's do, I guess black will be fine. We have to start separating, right? I'm drawing a line here. We're severing. We have to start separating chromosomes, right? Which means that the DNA here has to be split to put into the daughter cells, right? So those chromosomes have to start being separated. And like I said above, if we have damage to the DNA here, well, that means now we have damaged DNA here. And that damaged DNA is going to be replicated to the daughter cells, right? So that's why we have to have this checkpoint. We have to make sure everything is okay before we start replicating DNA, okay? So this DNA replication process, because you guys, this is not a molecular class, I just want you to kind of understand some of the basics. The DNA will open it like a zipper, and then we will start to read with DNA polymerases. You don't need to know what that is right now. Let me just get rid of this so you don't even need to know what it is. But these nucleotides here, we will make another strand of DNA here, right? You can see that those nucleotides will make another strand. And then the nucleotides here will begin to form another strand of DNA. And this is done by DNA polymerase. And then what we get is we get 2 new strands of DNA, right? So you can see there's one here. This is 1, and this is 2, right? And now we have separated the DNA into 2 separate strands of DNA, which can be placed into the daughter cell, okay? So again, don't worry about the molecular information. Just understand, all I'm asking you to understand here is that in this phase, we break the DNA into 2 equal parts. You can see here, we will open up a single strand from the mother DNA, right? We will open up here from the parent cell or the mother cell, right? Whatever you want to call it. And then that will be replicated to give to the daughter cells. They will each have their own strand of DNA. And that is why we have to have that checkpoint here. Because if anything is damaged previously, we will give that to the daughter cells and we will not have proper function of those daughter cells. Okay? So we can call this sister chromatids. Okay? And that just means that that genetic information is going to the sister cells. All right? So at this S phase, we will also have another checkpoint, right? So I'm going to just say right here, we will have a checkpoint because now another set of enzymes has to inspect if this DNA looks good, because we can stop the cell from moving further into the cell cycle at any point where there are checkpoints, and we can essentially kill the cell. We can tell the cell to undergo cell death, And that way we're not carrying that defective DNA into anything else. All right, so what's going to happen now is we will move to this S phase. We will have a checkpoint, and we will make sure that the DNA was accurately copied. We would make sure that the daughter cells have full set of genetic information. This should say now. Sorry about that. Now have a full set of genetic information. And we want to make sure that no mutations have occurred on either of these daughter cell DNA strands. Okay, so this checkpoint, we will say, okay, everything looks good. The daughter cell DNA looks good. We can now move to the next phase. Okay, so this is just simply another checkpoint. And that next phase is going to be, I apologize about that, we are going to move to the G2 phase. Okay, and the G2 phase is a lot like the G1 phase. We're going to have more cell growth because we're getting ready to split that mother cell in half, right? So we have this mother cell, right? Or a parent cell. We've increased all of the protein, right? We checked it. It was good. We've taken the parent cell DNA and we've created 2 equal strands of DNA. There's 1, there's 2, right? Okay, that looks good. DNA looks good. Okay, now we're getting ready to split this cell in half, right? Which is the mitosis process. But we got to grow the cell a tad bit more. And we got to just make sure that everything is checked before we split that cell in half. All right. So we're right here right now in this mitosis process. We're getting ready to split that parent cell or that mother cell into 2 daughter cells. So what's going to happen here? We're going to have this critical phase of growth before we start to split that cell, okay? And that cell is going to grow more. We are going to create mitochondria here, right? We have to put mitochondria into the daughter cells. We're going to double check, triple check, quadruple check our DNA, make sure there's no mutations, make sure that nothing is damaged, and then we can send that into the next phase, which is going to be that mitosis phase. Okay, so let me clear this really quick. I apologize. Let me just get that out of the way. That right there. Okay, so now when we enter this mitosis stage, okay, now the M phase is where the split occurs. Okay, so that mother cell, or we call it the parent cell, whatever you want to call it. I've seen it called both ways. Let me just put it there so that we're very comprehensive, right? Parent cell or mother cell, right? And again, these are satellite cells. That's what we're talking about. Okay, so this is a satellite cell. This is coming from the muscle. It splits in the M phase, and now we have 2 identical daughter cells. And those 2 identical daughter cells have genetic information that was split here in the S phase. It has all the organelles that were created here in the G1 phase. It grew more, it grew more in size, and it made more mitochondria in the G2 phase. All of the DNA was checked again. So in that M phase, now the cell splits, okay? And what happens here, we're almost done. Let me just close this out. Now what is going to happen is after that cell splits, it's going to do that splitting through a couple of subphases, and we don't need to worry about what the subphases are. I just put them here, but it's really heavy. And the subphases are basically telling us what is happening to the chromosome, right? What is happening to the DNA information, right? And I can show you what that subphase is. So watch, if I go here, right? Let me go back to this. I'd have to close it out. Anyways, when I showed you that picture of the threads, right, and the chromosomes that were kind of aligning themselves and splitting apart, that is the subphases. And I can give you a little more detail on that in a second. Let me just close this one out, okay? So now, after mitosis has occurred, if there's no more need for satellite cells, the satellite cells will go back to that rest phase and wait for the next injury, all right? And this is the cell cycle. And let me just kind of go back and show you those chromosome phases. Let me clean this up. So I just want to show you this here. So I told you that there are these subphases in mitosis, and you can see that here, right? This M phase contains all of these other phases. We are not going to worry about this at all. We just don't have the time, and this gets pretty heavy. And all this is telling you is that during these phases, this is when the most important aspects of mitosis occur. So this is where, this is the picture I was referring to. This is where those chromosomes start to separate, and they happen in these different phases. So that's all these subphases are telling you. So 1, 2, 3, 4. These subphases are what is occurring that eventually gets us from this to this, right? And we don't need to really worry about that because, like I said, it's very, very heavy. But I just want you to understand that the M phase, the mitosis phase, is the splitting of the cell, and it happens in the prophase, the metaphase, the anaphase, and the telophase. They're just small phases that are representing what is going on here. So again, not super, super relevant for what we need for this class, but I just want you to understand this cell cycle process. So let me see if there's anything else I want to punctuate, and then I will let you go. So that's all you need to know for the cell cycle. And again, these are in satellite cells, and those cells are waking up and they are separating and they are helping muscle regeneration and muscle repair. And we will talk more about this in class on Friday. I hope you all have a great day, and I will see you guys soon.